out today for our Fulbright Scholar Lecture. We are very honored to have Dr. Lakshmi here with us today to talk to us about her work with nanotechnology. As you may know, Senator Fulbright was a statesman for the 20th century who initiated and worked with different institutions across the world to bring in scholars at different colleges and universities so that we can share information. So while maybe you don't know a lot about nanotechnology or it's not your field of study or field of interest, I know that you'll learn a lot today by listening to this lecture because it impacts all of our disciplines, impact all of our lives. And so I want to thank you for coming out today, and I'm going to introduce Toya Dixon, who will introduce our speaker.
Hello. I just want to give some brief background on Dr. Lakshmi before I invite her up to speak. So Dr. Lakshmi is from India, having received her PhD from the Indian Institute of Science in Bengaluru in 2017. Her doctoral research was on the feasibility studies of magnetic nanomotors for biological applications. She's researched on the compatibility of magnetic nanomotors and various biofluids and developed conformal biocompatible coatings that can protect these nanomotors in bioenvironments. She worked towards engineering multifunctional nanomotors that can have great potential for theranostic applications, including magnetic hyperthermia. Lakshmi is currently working as a Fulbright postdoctoral research scholar under Professor Joseph Wang, the chair of nanoengineering at the University of California, San Diego, from 2018 to 2020, where she's working on the design of versatile micro nanomotors responsive to various actuation mechanisms and exploring their biological applications, taking them a step closer towards their future use as medical robots. Please join me in welcoming Dr. Lakshmi.
So good afternoon, everyone. Thank you very much to Toya and Tori for giving such a wonderful invite and welcome. So today I'll be talking to you mostly about the research that I have carried out during my PhD and some of the work that has been carried out in my lab at UCSD. So I am on a Fulbright postdoctoral fellowship for 2 years at UCSD, and I thought I would want to, but since Tori has already spoken about it, and since the deadline is in September, maybe I would not touch about the Fulbright program, so I would skip that. So for the next 40 minutes or so, I would be kind of taking you through the introduction of this field of micro nanomotors. We will look into the detail about the different types of micro and nanomotors that has been designed and fabricated over the years. And we will look into the feasibility of the magnetic motors, that is the magnetic nanomotors in particular, for the biological applications. And we will also look into some of the different applications that have been envisioned for these micro and nanomotors, and then look at some of the challenges of working with microrobots and conclude the talk. So about 60 years back, physics professor Richard Feynman had spoken about this idea of the surgical microrobots moving around in the bloodstream and acting to deliver drugs and also carry out some surgeries in the body. Now this kind of triggered a lot of interest in both scientists and non-scientists alike. And with the recent developments in the field of nanotechnology, there has been a lot of techniques that has been developed for the fabrication of these sort of micro nanomotors. So when you talk about the field of micro nanomotors, you're talking about artificial swimmers because you are fabricating them, and you would want to have an induced mobility. And mostly there has been a lot of envisionment for biomedical and non-biomedical applications, but I will be mostly touching on the biological part. Now, one of the main challenges of working with micro nano robots is you're talking about sizes which are in the micro nano regime, and this kind of leads to a more focus on the point of rho Reynolds number. So the rho Reynolds number or Reynolds number regime is the ratio of the inertial to viscous forces, and as we go down the size regime, you would have more viscous forces. So let's say if you are human, and if you're trying to swim in a pool of jelly, you will try to move, but you won't be able to move forward. So that's how it would be if you're talking about microorganisms at low Reynolds number regime. So you would have to come up with special strategies to move in such low Reynolds number regime. So if you think about bacteria, they would have the flagella, so it's like a corkscrew motion, which is kind of a main focus point for the design of the motors, which we would see later. And also like cilia, where you have the back and forth motion is not reciprocal, and as a result you can have motion at such Reynolds number. So the design of microrobots include various aspects. As the design, you would also have to think about how you want to move them, whether you want to power them by chemical or physical methods. You would want to see how you want to get them for different applications. And one of the main aspects is how compatible are they with the medical imaging technologies, and how they are going to move between the biological barriers and some of their medical applications. So coming to the different types of micro nano motors, the micromotors are broadly classified based on the actuation mechanism into the variety of stuff, but we can kind of group them as the ones which are actuated by the chemical methods and the ones which are actuated by the physical methods. The chemical methods are those in which the micromotors use a chemical fuel like hydrogen peroxide or even acids, and that kind of can cause some release of bubbles which can cause the motion, or it can be electrophoretic in nature. Now, if you're coming about the physical methods, it can be either driven by the ultrasound or the magnetic fields, which is what I will be focusing on because my PhD was on that. Now, one of the main concerns for using the chemical motors is, since you can use hydrogen peroxide for experiments in lab and stuff, but would you want to add hydrogen peroxide in the body for biological applications? So the answer is no. So you would want to do something which uses no external chemical fuel at the same time which is safe to work with. So let's look at some of the examples of the chemically powered motors. So the first video what you see are actually magnesium particles, and they are actually moving because of the evolution of hydrogen bubbles because of the seawater. And the second one what you see here are zinc micro rockets, and the zinc actually reacts at acidic pH and leads to the creation of hydrogen bubbles which kind of cause it motion. So these are some of the motors, but you have a variety of those which can move by acid, even water, bicarbonates, etc., depending upon what is the material of your micromotors. Now coming to the physical motors, the first one what you see is actually the motors which are powered by the ultrasound waves, and the second one, which is what is actually the one that is helical in nature, but it is moving with the help of magnetic fields. You also have a variety of hybrid motors, that is motors which are powered by both of them. That is, you would have a magnetic film attached to one part of the motor. You would also have the ultrasound, that is a gold film segment attached. So you can either actuate it by using ultrasound or magnetic, chemical and light, magnetic and chemical, and all sort of variations. So depending upon what applications you would want to go, you can explore the different combinations. So coming to the feasibility of magnetic micromotors, there has been a lot of envisionment of applications for the micromotors, but we have not really achieved too much. So one of the main issue is, like as I discussed, with the case of chemical micromotors, they are basically limited to the in vitro applications, and also the material what we use for the fabrication of these micromotors have to be biocompatible. So biotoxicity is one point we would have to really look at. And even though people have shown the motion of these motors in biological environment, no one has really studied the long-term stability, that is how hazardous they are to the biological system. And here you have two figures from papers wherein people have tried to see how these motors can move in blood, and they have seen that if you dilute the blood like 1,000 times, or if you take the blood to the physiological temperature but the viscosity is less, you can have some amount of motion. But no one has really showcased a motion of a micromotor at the human blood concentration. So here you're talking about, like, say few thousand cells or even 10,000 cells per ml or something, but like you would have in 1 microliter of blood you have around 10⁻⁶ number of cells. So if you dilute the cells, you can get some motion of these motors, but if you really take a human blood in general, you won't be able to see anything. So coming to the fabrication of the nanomotors, so we actually call them as the nanopropellers. We take a layer of polystyrene beads, that's the SM of the polystyrene beads. We kind of edge them down to reduce the size and have spacing between the beads. We use a physical vapor technique called as glancing angle deposition, that's called GLAD. So what it does is you have the substrate attached to the top. You have the material of interest which is getting evaporated from the bottom. So you keep your substrate like this. So the polystyrene beads are like this. The vapors come and attach to your polystyrene beads. At the same time, the substrate is rotated at regular intervals. So that is what actually causes the fabrication of these structures. That's why they're helical in nature. So once that is achieved, we would sonicate them out, put them on a silicon wafer, and do a magnetic film deposition. Once that is achieved, our samples are ready for experimentation. We sonicate them out in water or biofluids of interest and use a Helmholtz coil. So what you see is a 3-dimensional Helmholtz coil. So this is what actually gives it the rotating magnetic field. So if you take about a screw, what happens is if you're going to rotate it, it's going to move forward. So same mechanism works here. You have a rotation translation coupling. So as you apply a rotating magnetic field, you're able to move the helices forward. So here you have a movie of a helix that's moving in the magnetic propeller moving in DI water, and as you can see, it can move back and forth, and we have designed the system such that it can move in all the full 360 degrees, like you can have the control in the directionality of these motors. Now what happens when we try to put these sort of nanomotors in biological fluid? So it started off as a one evening experiment where I took some phosphate buffer saline, which is something which is very close to our biological system, and what I saw is when you put these motors in biofluids, they don't respond at all. So what started off as a one evening experiment, it took me almost 1 year to figure out how to come about. So that was the starting of my PhD. So we found out that what we found out from our experiments are the iron or cobalt which was put over the helices, they were actually getting etched or getting corroded in the PBS because it has higher concentration of chlorides and phosphates, and that can react with the cobalt and iron over the helices and can cause them being non-magnetic. So again, cobalt is not a great material for biological experiments because of their toxicity issues, so that's something we would have to see. And if we want to move these nanomotors in a system like blood, it would need enough thrust to move between the cells. So these were some of the challenges we would have to look at. So for my experiments, we looked at phosphate buffer saline, simulated body fluids, FBS, that's an addition into the cell culture media, and finally the blood. So we took our helices coated with iron or cobalt. Then we tried a variety of conformal coatings, that is, you're kind of draping these helices with different materials to really protect the underlying magnetic layer inside so that they don't react with the biofluid. At the same time, their properties also okay, like they are biocompatible in nature. So we looked at a thin coating and also a thick coating. So we also, we did the experiment wherein we immediately sonicated these motors down in the biofluid and tried to see how they're responding to the field. We also put them in for overnight incubation to see after a day or two of incubation if they are still able to respond to the magnetic field. So from our experiments, what we saw is there's a material called as zinc ferrite. So these are the SEM images of the swimmers before and after. And as you can see, after the coating of around 200 nanometer of zinc ferrite, the structure, the rigidity is still maintained. At the same time, the swimming performance is not really changed too much, so the material seems to be okay. And we were able to actually move these nanomotors in blood. So this is about 1.8 X dilution. So you would ask why you have a slight amount of dilution. So the point is we would have to sonicate these nanomotors in saline and then put them in blood because we cannot really sonicate the blood cells. They are going to disrupt. So that is one of the reasons. So we are now trying to see whether we can, instead of how do we release the nanomotors at negligible dilutions. So this was the first report of a successful voyage of the nanomotors in human blood, because people still report like 100 or 200x dilution, but this is the closest that they have gotten to the negligible dilution of human blood. So following this, we carried out the biocompatibility studies wherein we took these nanomotors and tried to grow some cells. So we used the mouse myocyte cells, and we did the MTT assay, which is basically a toxicity assay. So you grow the cells over these structures, and over the period of 3 days you would want the cells to grow. That is, if there's no toxicity, the cells would grow over these structures. If the material what we have over these motors are toxic, the cells would not grow. So we do an MTT assay, and we saw that the ferrite coating and the iron with ferrite coating was good enough for our experiments. And as we know, the cobalt is not a great material, and the cells did not really grow, like even after a period of 3 days. And even ferrite coating did not help much. One of the reasons is that the ferrite coating, you can have some cracks here and there, and that crack is good enough to basically leach out the cobalt into the medium and cause the toxicity to the cells. So from our experiments, we see that we did some fluorescence imaging and also some SEM images to reiterate the point, but we could see that the zinc ferrite coating was biocompatible for our experiments. So another aspect of our experiment, what we did is to see whether we can really study the motion of these nanomotors in blood. So as we see, we could have two scenarios, whereas the bottom one, you can see that the motors are able to move from one position to another, whereas the top part, they don't really move much. Like, it's rotating, but it's not really getting translated. So one of the reasons for that is, like, because we are doing these experiments on a glass slide setup, because it's still in the lab system, once the nanomotors get to the bottom part of the slide, they are not really able to move around because they are blocked by the cells from all sides. So we have kind of focused on the high-speed scenario. That's the second movie. And we also looked at what are the different motion that the nanopropellers would have in blood. So one of them is, because of the concave nature of blood, the motors would kind of get stuck. If you see the second movie at the bottom, the motors would kind of get stuck to the concave part of the blood cells, and after some time, they would kind of come out. The top movie actually shows that you have a lot of cells. So when the cells, because they're dynamic in nature, they are going to come close, and the motors, they will try to move through the cells, but they can't. But again, with the rearrangement of cells, they are able to move through. So we kind of looked at the motion of these nanomotors. We actually call the period where it doesn't move through as the jamming period. So we try to see what are the different aspects which would govern the colloidal jamming. So one is, we have tried to look at the size regime, the effect of plasma, and the density and stiffness of cells. So I'll be talking about the density and stiffness of cells. So just to give an idea, we try to look at motors which are 2 microns in length, 3 microns and 5 microns in length. And we see that 3 microns, which are the movies what you saw so far, are better for motion in blood. And talking about plasma, plasma does not really have a role in the colloidal jamming because there is no cells present, and the viscosity does not really affect the colloidal jamming. And talking about the density and stiffness of cells, we actually took the the concentrated blood, and added volumes of saline, like depending upon if we want to work with 1x, 2x, 3x, and etc. And then try to study the motion of these nanomotors in blood. So these are all the graphs, but the point is, what we see is, as we increase the dilution, we would have more, as you would expect, like less number of cells, so you would have more motion of the nanomotors. So that's one aspect we see. And another point is, we try to see what is the effect of stiffness of the cells on the motion in blood. So what we have taken is, we have taken the blood, removed the plasma, so we are just left with the cells, and we treat the cells with glutaraldehyde. So glutaraldehyde is a cross-linking agent, so it makes the cells rigid, and we do our experiments in a matter of 2 hours. So 2 hours is something where you expect the membrane stiffness to be maintained. So we have done all these experiments from the blood of lab, my friend, so that we don't change any of the parameters. So he was the guinea pig, but it really worked in my favor. So again, I'm just not going here. So okay, just to look at, let's look at the, So this is the 3x dilution. So one thing what you can see is, as you increase the dilution, as you increase the stiffness, the cells do not really move much. They are kind of, you can expect, because if the cells are very rigid, they won't be able to push and move through. So we did it for a variety of concentrations of cells, and also the glutaraldehyde treatment. So we see that as the cell stiffness increases, the TU, that is the time of unjamming, that goes down. That is, they are jammed for most of the time. So this is just the velocity. So what our finding was, that we could try to use them as a rheological probe for finding out the properties of blood, because for certain medical conditions, you would have, for example, in the case of diabetes, you would have the cell stiffening. So that's something we could try to use these as rheological probe for blood cells. And these are some of the applications which we had tried to look at for our nanopropellers. So we looked at the stability and toxicity, and now we will go into the, we could say the rheological probe for some extent, and we will go into the magnetic hypothermia. So the zinc ferrite coating, which I spoke about few slides before, they actually are known to be having this magnetic hypothermia property. That is nothing. When you apply a field and frequency in the range of kilohertz regime and a high field strength, you are basically making this material go through the hysteresis curve number of times, and that actually leads to a heat generation, and that can actually cause the heating of the environment. So what we have seen from reports is if we are able to heat up the cells from, let's say, the physiological temperature of 37, which is where you normally incubate the cells, all the way up to 43, 44 degrees Celsius, you can actually cause some cell death. So we try to explore this concept. So for this experiments, what we have done is, instead of having a thin film of zinc ferrite, we have kind of draped the nanoswimmers from all sides by changing the recipe for our zinc ferrite synthesis. So if you see, this is the before just the silica nanoswimmers, and this is the one after the zinc ferrite. So you have a zinc ferrite coating from all sides, and we did some characterization of the zinc ferrite using EDS, where you can see the zinc and iron peaks, XRD, which can kind of give you the structure, the peaks which are related to the zinc ferrite. And we also did an XPS to see what are the elements and where do you get peaks to finally confirm it's zinc ferrite for sure. And from a hypothermia experiment, so what we did is we took our nanoswimmers and put them with the HeLa cells, and we used the field and frequency in the range of kilohertz for 20 minutes, and after doing that we do the MTT assay to see what sort of cell death we are getting at the end of the magnetic hypothermia. So one thing what we see is that if we use a 2 milligram per ml concentration of ferrite for our experiments, we are able to achieve a cell death of around 50%, and this is just the cells before the hypothermia treatment, where all the arrows, I don't know if you could see, they are pointing to the magnetic nanoswimmers and the cells look fine. And these are the images after the magnetic hypothermia, where you don't see much cells because the cells have died and they have got dislodged from the plate. And also you could also see some amount of, you don't see the cell structure, the cell has actually ruptured. And we could see from experiments that the zinc ferrite coated nanoswimmers could be used as magnetic hypothermia agents. And this is one of the experiments that has been done in our lab, wherein we have incubated these nanoswimmers inside HeLa cells and then try to move them inside the cells. So here we are trying to write a letter N inside the cell. Yeah, and so we try to write N and M like nanomotor. So we have the sort of control over what we want to do with these nanomotors. And we have also tried to see inside the cells what is the rheology, like how viscous they are and stuff. So that's something which is being carried out for now. And another one is the rheological measurements. That is, depending upon, like here we are showing that you are having the water system and you have the glycerol system. So depending upon what is the viscosity of the media, the motor would move differently. Like it's a little physics related, but instead of doing a proper motion, they would have a change in the motion, and looking at what is the angle in that sort of motion, we can correlate and find out the viscosity. And this is we have taken a glass slide with water on one side and glycerol on one side. So we have taken this nanomotor from point A to the point B, where you just have the glycerol. And what we have seen is, as you have moved from one point to the other, you actually have the viscosity change and also the change in the motion of the nanomotor. So we have done it for different systems, and it has really been successful. So that's some of the applications we have explored the nanomotors, because the lab in which I did PhD is more like a physics lab, so we look more for the rheology and physics related applications. And coming to the applications of different types of micro nanomotors. So the lab which I am currently based in, we are basically a electrochemical chemistry based lab. So we kind of do everything using chemicals, not exactly chemicals, using like biofluids or acids and stuff. And we fabricate them using electrochemical template deposition, that is just electroplating. And I will show you some of the interesting works that we have published from this lab recently. So some of the other applications of these nanomotors is, one is the pH sensing. That is, if you have this nanomotor coated with a pH sensitive material, you could have some color change. Here you have heavy metal sensing, that is, you can take this. So this is actually quantum dot decorated nanotubes, and if you have heavy metal like mercury in your system, the mercury can go and attach to your motors and can quench the fluorescence. So that's one application we have shown. Another one is the oil removal. So I will go into that in detail later. And some of the other biomedical applications is biomolecule sensing, trying to drill into a cancer cell, trying to have some drug delivery. Again, I'll go some of them into detail later. So this is basically a nanomotor which are powered by a little bit of hydrogen peroxide, and it has been modified by lectin, which is a protein. So it basically picks up the E. coli in the medium. You have a lot of yeast cells, but also E. coli. So you're basically taking them to the E. coli part, and it gets picked up by the motor, and it moves forward. So you can actually have a selective picking up of these cells of interest depending upon what molecule you are getting attached, modifying your motors with. And the next one is you have oil removal. So here what you see is a nanomotor which is basically carrying a lot of oil droplets. So the surface of these nanomotors have been modified with thiols. So thiols are hydrophobic in nature, and those would be attracted to the oil molecules. So this is some of the applications people try to look at for, like say for oil spills or oil decontamination. If we could use, can we deploy these nanomotors for a larger purpose, something like oil decontamination? So that's something we have shown. And coming to the ultrasound propelled nanomotors, what we have done is we have taken the gold nanowires and then modified them with a thiol group and added CRISPR complex, that's a Cas9 sgRNA. And the one good thing about ultrasound is if you have these gold nanorods and the cells together, the ultrasound waves can actually cause the penetration of these gold nanorods into the cells. So here we are talking about the cancer cells, and cancer cells have a higher expression of glutathione, and that would cause the release of these complex from the cells, and you would have the gene. So here we are talking about gene knockout. So you're talking the cells where you have the GFP, that's the green fluorescent protein, which is expressed very highly in these cells. And after the ultrasound actuation and we do the incubation for 2 hours, we see that the green fluorescence has been quenched in these cells. So when we do experiments in lab, we would have to do this silencing for a period of 2 days or more, and here we have been able to achieve them in a period of 2 hours. So ultrasound based motors could be something which we could look at in the future for our experiments. So this is just some of the data from our study. So what you see here is the motors which have been modified with the complex just moving in the ultrasound waves. So here you have a lot of mixing around, and when you add the cells, you are doing an experiment for around 5 minutes or so, and you are still able to get this the complex inside the cell and get their gene knockout done. Now coming to another aspect is the acoustic microcannons. So for delivering drug to the body, you would have some patch. So here what you have done is you have these motors, and so the motors are filled with the drug molecule of interest, and also we have a chemical called as perfluorocarbons. So what happens is at using ultrasound waves, the perfluorocarbons, they would actually vaporize, and that would cause the bursting of these drug molecules from the surface. So that's what is shown here. So here if you see, you have the SEM image of the microcannons wherein you have the particles inside, and after the ultrasound actuation, you see the particles have been released from the surface. So that's something we are trying to look at, if we can use this sort of system for delivery of drug on the epidermis. Moving forward, some of the biomedical applications that we have looked at is using the acid in our body to move these nanomotors, so you don't have to depend on any chemical fuels. And also one of the other aspects is to use a biologically biohybrid motors, where you have the nanomotors coated with membrane fragments like RBC or platelets, so that the system is mostly not really immunogenic, that it doesn't really cause any immune response in the body. And the ultrasound, we have already discussed about the sensing part. So talking about the nanomotors in the gastrointestinal tract. As we know, in the stomach we have a very high acidic pH. So the acid is good enough to actually cause, if you have motors which are made up of zinc or magnesium, the magnesium or zinc would react and form Zn2 plus or Mg2 plus, and would also cause the release or the evolution of hydrogen bubbles. And those hydrogen bubbles are good enough to cause the motion of these nanomotors. And also in the case of magnesium, you can see that magnesium, even in the presence of water, could also lead to hydrogen evolution, just that the rate of hydrogen evolution is a little bit on the slower side. So here is actually a movie of the P dot, which is a polymer and polymer and zinc micromotors in the gastric acid. So the zinc would react with the gastric acid and cause the evolution of hydrogen bubbles, which is what causes their fast motion. And what we have taken is, we have taken these zinc-based micromotors and tried to do the experiments in rat models. Like we have taken rat and put the motors inside the mouth, like we have given as a oral gavage. And we have seen, we have taken the toxicity studies and tried to see what is the sort, like these motors are kind of functionalized with fluorophores. So you would actually look at the fluorescence imaging of the organs of the, or stomach of the mice and see where they are distributed. So that was one of the starting point of our research. So we have taken, so there's something called as enteric coating. So enteric coating is something that is based, it's a polymer, and based on the pH of the environment, they are going to dissolve, and as a result can cause the delivery of whatever is inside. So here we have taken a, we can say a micromotor which has magnesium inside. You have a P dot, and on top of that we have an enteric coating. So what happens is the motor, let's say you are giving it to the mice. Once it reaches the, so this enteric coating is sensitive to pH at 6 or 7. So it will be kind of it will not get dissolved at the acidic pH of the stomach, but when it reaches the gastrointestinal tract, that's where the enteric coating is going to get dissolved, and you would have the release of the molecule of interest. So here these are actually the gastrointestine of the mice, and as we see here, you have these are the regions where you have the motors after 6 hours of orally administering the motors, and this is after 12 hours. So even after 12 hours, you have some amount of retainment of these nanomotors in the intestine. So that's something we would want to explore more with regard to the drugs part. So this is just a video which shows how you're talking about the administration of these enteric coated micromotors. So you have the pH 1 to 3, wherein it's very highly acidic in nature. You have the nanomotors moving, and they don't get dissolved, and as it reaches the 6 to 7 pH, the enteric coating is going to get dissolved at that pH and would lead to the release of the motors from the inside. Now coming to another research aspect is, can we put these nanomotors in pills and try to administer them? So we have taken these nanomotors, in this case the magnesium motors, and kind of added the lactose and maltose, which are the excipients, in a pill, and mixed them up and made pills out of them. So these are the pills that we have fabricated in lab of different sizes and shapes, and we have administered these pills to the mice and tried to see how they are distributed. And these are the stomach of the mice, and as we see here, we can see, this is a free motor and the motor pill. So in the case of motors which are in pill, you have a lot of concentration of these motors in the stomach region, which is what we were looking at. So that's something we are in studying more. Another one is, can we use these micromotors as for vaccination purposes? So we have taken these magnesium motors and conjugated them with the antigen of interest, and then given them enteric coating again and administered them to the mice and try to see how is the antigen expression after a period of a month or so. And we see that the values are pretty, this is the case of the motor toxoid, and we have got a pretty high toxoid expression in the mice which have been given this compared to the normal or the static motors. So that's one application we are looking at, and we are also in the process of designing a more multi-compartment based motors where you can have a drug or two drugs together, wherein based upon the stimuli you can have the release of the drug for various applications. And talking about the hybrid biomotors, this is something what we are currently doing, that is you have a magnesium motor and we try to attach, in this case that's the macrophage. So that's basically a cell which you have in your body which fights against diseases or infections. So in this case you have the magnesium motor with the cell attached, and we have tried to see if it can be used for detoxification purposes, that is to pick up the toxoid in the system, and so that is toxoid in the system, so that's something, and if it can neutralize, because the macrophage is known to neutralize these in the body. So if it is attached to a micromotor, can it do that? So from our experiments we see that yes, over a period of even 10 minutes we can see that the LPS, that is the toxoid in the media, has come down. So that's something we have looked at, and more work is on the way. Just to this another work, but the point here is the magnesium motor actually uses the pH, the acid inside the body or in the stomach to basically cause their motion, and since you're using up a lot of acid, it can kind of neutralize the pH in the area for some time. So if you talk about all the anti-acid you would take for the PPIs, something which the tablets you would take, so these sort of motors are kind of acting in that direction. So this is basically the stomach of the mice. What we have seen here, this is just in water where you don't have, it's just how it's the acidic pH. Now at 20 minutes after introduction of the motors with the magnesium motors, you see that the pH has kind of neutralized. So this color, the dye which is attached to the motors was actually a pH indicator, so that's why it shows that the pH is not same as here. And after 24 hours of administration we see that it's similar, it's back to the normal. So we are having a prolonged effect of these motors in body. So there's some application which we could look at. And one of the other application what we have seen is we have taken these motors and attached antibiotics and try to see if it's good enough to reduce the treatment of stomach infection, in this case pylori, Helicobacter pylori, and we have seen from experiments that after a period of 1 month the number of colony forming units inside the mice has come down compared to the other controls. So that's some of the applications which we are looking at. Another one is using the ultrasound waves. Again, it's a neutralization technique, but here you have a nano rod with again membrane attached, that you have the platelet as well as the RBC membranes attached, and you add the molecule of interest and do the experiment. So it's basically a neutralization of the pathogens in the system. So coming to the challenges and the conclusion. So there have been a lot of enrichment of these micromotors for various applications, but we also need to look at how much we can deliver, that is if we want to move from an animal model to a human system, what are the different scenarios we would look at. Let's say, do you want all of them moving in the same direction? What is the long-term effects, what are the toxicity effect of these motors in body, let's say after a month or two, what are the materials of interest, how can we move these motors from a remote position, do we need to use a chemically powered motors or even or do we need to go for let's say a physically powered motor. So the problem with ultrasound, even though we focus on all the good experiments, is we cannot really generate for so for ultrasound to work in a system you would have to create nodes, but we cannot create that nodes in the human body. So that's one of the main limitation of ultrasound system, which is where the magnetic fields would work, because you can remotely work on it, and let's say you have a coil or something, like how in MRI you have the magnetic field all over you. So that's something we could look at. So that's where the magnetic powered micromotors would have upper hand over the ultrasound. And coming to the conclusion, so we discussed about a variety of nanomotors and some of the various applications, and there has been some, I think 1 or 2 groups where they have kind of working on the paperwork to move from the animal studies, let's say from mice to pig, and try to move forward from there, because a lot of paperwork for such work, because of the various aspects. So one of the main focus should be on the long-term toxicity studies and to see how they work when and how toxic or what are side effects affected. So this started off as a dream of Professor Richard Feynman's, and hopefully with all the coordinated push we could usher into an area of non-invasive therapies in the decade. So to conclude my talk, I would like to thank the faculty members and the organizers of Forsey Technical Community College for giving me an opportunity to present my work here. And special thanks to Professor Joseph Wang, my host advisor at UCSD, who has been kind enough to share my details here. And my PhD advisor, Ambarish Kose at Institute of Science, who has been very supportive. And my colleagues at UCSD and IAC, my Fulbright Fellowship for giving me an opportunity to come to US and pursue my research. Then IAE for the Outreach Lecture Fund, which kind of got me over here, and friends and family. And I would also like to thank Shiva for taking his time out and coming all the way here and supporting me for the same. Right, thank you so much.
So we're going to open up for question and answer. And my first question, Lakshmi,
Sure.
is, you know, the work that you're doing is fascinating. But even though we have a nanotechnology program here, the majority of our students come in with no clue, no idea what nanotechnology is.
Yeah.
You know, they need to have their Google ready to
Right,
follow
right.
the conversation. How would you introduce the work that you do, or how should we talk to students about nanotechnology to get them encouraged in layman's terms that would apply to the average person?
Oh, that's a good question, actually, because my background during graduation was in biotechnology, and I thought, okay, I did not want to really get into the molecular aspect. I was not very interested. I was more scared, I would say, about the molecular biology part, and that's when I came across this course of nanomedical science, and I thought, okay, let me just give it a shot, and I applied for the entrance, and I got inducted into the program. And once after getting inducted, I was all over the place because you don't know what you're getting into, because there is not a good fresher course wherein you say like this is this and that is that. So the introduction was always difficult, and since it was a postgraduate course, it was still better but not the best. So I think it's always difficult for people to start off with nano, and we have courses like nanophysics, nanochemistry, which makes it even more difficult, because coming from a biotechnology aspect, from a background, you do not have basic course in physics or chemistry during your undergrad, and directly being thrown at not even physics but nanophysics is like quantum physics. That's like a nightmare. Yeah, Schrodinger's equation still kills me, so so that's a nightmare. So I would say maybe you could get people in, I would say, because the work, okay, let's say the work which I have done in my PhD, because I am from a biology background, and I have shown more of the application part, it looks more interesting to the public. But let's say if I have shown the real, like the derivation and the theory and everything, no one is going to get interested. So I guess, yeah. So I guess if you kind of show people, like to start off with wherein, let's say you have something like you go from a micro to a nano, and kind of fascinate them, like these are the things you could do, and then try to go back to the background, like you have to start with physics and chemistry. I think the first initial push towards nano should be, I think, more visually rather than just the courses. It's going to be very hard. That's from my perspective, I think.
I agree.
Yeah.
Thank you.
All right.
Great
Thank you.
lecture.
Thank
Really enjoyed it. A couple questions
Sure.
that just intrigued me. One is, what is the size? I think I saw it up there of your Nano Motors. Are they 5 micromotors?
Yes, sorry, I should have added on this. So the width of the motors would be around 506 nanometer, and the length would be around, we could tune it as how much we want to deposit. So I have worked with 2 micron length, 3 micron length, and 5 micron length. But right now, the people in my lab, we work with something less than 2 micron, because when you're doing cell incubation experiments and stuff, it has to be smaller than 2 microns, else the structures cannot be taken up by the cells.
Okay. Sorry, just one last question.
Sure.
That is, when you talked about the aspects of drug delivery, has any animal work been done with drug delivery in the bloodstream?
No, so my PhD work, we started off with the blood and stuff, and I have done few experiments, I won't say all, few experiments to see, because all of the flows what I show here are all the static flow, never the dynamic. So we kind of procured a syringe pump and added the motors with the blood and try to see how they move. Now, one of the main issue is like the flow in blood is like talking about meters or centimeters per second. So in capillaries, the velocity would be in centimeters per second, or something close to that, or even millimeters. Now the lowest what we could achieve with a syringe pump is even limited, but even with such flow systems, you cannot really get them moving. Like it's just going to like, it will just move. Like you pass blood, they are going to go left, right, and center. They cannot really move against the flow of blood. Another thing is they can actually get attached to the blood vessels on the side of the blood vessels and kind of get lodged over there. So how do we, they always tend to move closer to the walls rather than be in the midstream. So that's something, that's one of the reasons why you see people working in, like you have papers or research happening in the eye and other regions where it's basically a static flow, you don't really interact with blood, but no one has really worked on those parts. So that's a important aspect, yeah, sure.
How long does it take for the particle to disintegrate? Because it can't stay there forever.
No, so if you're talking about the pills or the magnesium motors,
So
sorry.
whatever you're going to do, whatever motor, whatever...
So if you're talking the motors in the pills, you're talking about, let's say, depending upon what excipients you add, it would be dissolved in 5 to 10 minutes. And if you're talking about the magnesium motors, the hydrogen of evolution bubbles and the full consumption of magnesium waters would again be over in 5 to 10 minutes. Again, depending upon what acidity you work with, but if you're talking about a proper gastric fluid, it should be done within 10 minutes.
So the question is, why do you want to move it through the blood as opposed to attaching it on the blood and let it move with the blood? Because I see, because the viscosity of the blood changes if you're dehydrated, different factors
Yes,
like that.
yeah.
So why try to move it through as opposed to attaching it onto the blood and it moves, and then when it gets into maybe the interstitial fluid or whatever, then it doesn't work?
Yeah, we could do that. I mean, just that I guess people have not really worked on it, and I guess the kind of sophistication you would need, I mean, because when you're talking about mice also, you would need a proper magnetic system. Okay, let's talk about the magnetic system. You would need a proper setup to work with where inside the coil the mice should be able to fit in and all those. So you would need to have a generation of higher field and frequency, but no one has really explored this, but yes, because we always talk about flowing against the blood, but why can't it move in the direction of blood, right? Yeah. Yeah, I guess no one has explored it, but for sure, yes.
The second question, you talked a lot about the stomach and everything else like that. This will also be applicable for the blood-brain barrier for strokes and stuff like that. So
Yes.
are there plans to do something like that?
In my lab or in both the labs I work with, I have never, there has been one recent research paper wherein people have tried to take a excised brain of mice and try to inject the motors into one of the parts, and they have shown some motion in using it was not really a living mice, it's just excised brain, and they have shown the motion, but that's the only thing I have seen with. And with motors, because we always talk about the blood-brain barrier, but I don't think anyone has really researched on it. There has been another paper about magnetic nanoparticles wherein you kind of spray the motors through the nose, and they have seen that it's distributed over the brain and stuff, but not really. No one has explored the brain aspect much.
So I think that would be pretty applicable.
Yes.
Because then when you have a stroke and it bursts and the blood bleeds out and stuff like that, delivering like ATP and stuff like that. That
Right,
would be something
right, sure.
unique. Or you could just drill a hole and put it in. As opposed to the blood-brain barrier.
Yeah,
That's
sure.
a mechanism
Yeah, sure.
that you could
Right.
I have a couple more, but
We can do this all day. You know, well, just bridging on Sheldon's question, what about this cerebral spinal fluid?
No one. I mean, people have shown the motion of simulated cerebral spinal fluid, but no one has really explored. I mean, this field has really come in. At least the in vivo experiments would have come, I would say, maybe like 3 years before. I mean, from now. So it's a very infancy stage. I mean, because you do experiment, there are cases where you see that the mice die immediately after administration. The experiment is stopped there. You start fresh with the new motors and stuff. So getting there is a. So I can tell you, like, there's one of my colleagues who works in Johns Hopkins University. So they are working on grippers for using the motors for tissue biopsy. So they started off like it pretty well, and they procured the pigs for the experiment. So procuring pig, I mean, it involves a lot of money, and you had to kind of feed them. It's even messier than mice. And they got 2 pigs, and the 2 pigs died on the operating table. So they stopped the pig, and they have gone back to the mice. So there are a lot of, the issues that comes with doing such experiments.
big grants.
Yeah, that's what.
Also, you could use it for missing person, like in the ocean and stuff like that. I think that's also another application that I saw. That because I don't.
I'm listening.
Oh, you're listening. I can talk to her. Yeah, because missing persons in the ocean, you could send those nano
Motors.
and then
Yeah, more like intelligence
Yeah.
motors, yes. We could, I mean, just that people try to look for, I won't say eye-catching applications, but we try to look at more the biomedical and, chemistry and physics or bio-related. People don't really look for the real scenario. Maybe that's the reason, but I have never heard anyone talk. But it's a good point. We could.
So that said, who are the major, what areas of major are funding this kind of research? Because I know that can certainly direct the direction of what we tend to look at.
I would say chemistry is very helpful. Again, depends upon what sort of motors you want to work with. For example, the lab where I did my PhD, it's basically a physics lab. So for me to fit in was a little, not difficult, but my professor was very supportive that he was like, okay, you do whatever you can. And because I don't think he expected me to sit and derive stuff and even do some computation, which was not my area of interest. So okay, there are people in my lab who has physics background and they do like wonderful simulation work, computation to see, because one of the main aspects is you say this is what it does. You do the viscosity, you do the rheological probe, but does it really match with that of the theory? So that's one thing what is very nice. So physics is very good. Again, electronic and communication is very good, because if you want to have a wireless communication with these nanomotors, you would want a good background in EC, and again instrumentation, because the engineering of these parts, that is the amplifiers, making the coil and everything, you would need some amount of background in that particular aspect. Chemistry for sure, yes, because if you want to modify the motors with different molecules, or even make them, like say for imaging, fluorescent, or make them more biocompatible, less biocompatible. Even material science is good, because the lab which I work with right now, it's mostly a chemistry lab, and material science is very good in the sense that if you have a good knowledge of what material can react in what sort of media, like acidic or basic, you could use those materials for your experiments. So I guess even if you have physics or chemistry or biology or materials or EC, you could still work in this field. So that's the good and the bad thing about the interdisciplinary, because you learn new things, but you could also make use of whatever skills you have so far. Because my background is not chemistry, but I think I'm doing decently okay in my present lab.
So you mentioned interest in biomedical, so pharmaceutical companies will probably be looking at this, and you also mentioned like using the nanomotors for oil.
Yeah.
So maybe the
Oil
petroleum
spills,
industry.
yes.
What other industries would be looking at this material?
I think cosmetics, in the sense that in my lab in India, we had a collaboration with L'Oreal where they wanted us to use these nanomotors to see the viscosity changes in some of their products. Like they wanted to see whether we could use them and try to, kind of see what is the viscosity at different levels, because for cosmetic industry, the viscosity is very important. So that's one aspect we could look into. Another one, here I know that we have some collaboration with Novartis. They are working on those aspects, some of the collaborative work.
Agriculture?
Yes, why not? Yes. Because it's a very open field, right? And people try to, like, we are showing some toxoid or something for diseases. People could use that for even treating animals, because all our experiments are done with animals. So these are all I could think of so far.
My next question was, particularly here in America, we have a lot of hard time attracting women and minorities to the STEM fields. What have been your experience as a woman of color in the STEM field, and what could we do as practitioners to bring in other women and minorities?
Okay. Okay, to US. Okay. In general. Okay, so that's a good question. So I got married when I was in my post-graduation, and I had to really get everyone. So I was very happy when I got selected for my PhD program, but none of my family was. They were like, what's going to happen to your family life? What about kids? Because in India, the main problem is you get married, the next month they would ask about the good news. So that's always the pressure. So that's always the pressure on our heads. Yeah. So I get into the PhD program, and people always, so I kind of tried avoid going to any functions, because all the eyes were like, they either, rather than looking at my face, me on my tummy. So 2 years or 3 years went by, and I had this big exam. We have this comprehensive exam that kind of confirms that you could continue your PhD. So that's when everyone was looking forward to, and I got pregnant a few months after that, and I had the baby during my PhD. So I had the support from my mother, but it was not easy. So finishing PhD itself was not a pain, but it was a lot of stress on my head and everyone. And one of the main thing is people don't really appreciate that you have finished your PhD. People always think it's your partner who has been supportive, but it's you who does that. So I think you need a lot of support system to really say that, okay, you have done the work, so it's kudos to you. I mean, they do require the congrats and everything, but you should also acknowledge that you have taken your time off, try to maintain both the family life and personal life, and try to make it work. And then after my PhD, again, the problem is the location constraints, because I cannot move away, let's say 20 or 30 miles away from where I'm staying, because my family is around. So that's, again, added pressure. And then I applied for the Fulbright program, and I got selected. So that was even worse, because you are moving to another country. So what will, again, happen to your family life? So I moved in here with my daughter. My husband is staying back in India. So it adds a lot of stress on my family life, for sure. It's not easy, but I guess you would have to do what you want to do. I mean, you cannot have everything together. Like, I can maybe sit at home, I mean, not sit at home, like try, be in India and try to explore whatever the limited options I have, but I could use these 2 years as a stepping stone where next year I go back and I can have better academic career prospects. So I think the support system is very important, and I would say family has to be very supportive. People has to, I mean, I won't say you need motivation every day, but you would need every other day to say that, you know, you're doing okay, it's fine. Maybe you are not able to, feed your daughter once, 3 times a day, but if you're able to do 2 times a day, it's good enough. So that blame, I think that should never be there. And I think that way coming to US has been great for me, in the sense, when I was in India, you had a lot of restrictions put on you, like, you know, don't do this, don't do that, and everything. So here I am able to breathe more and kind of explore my opportunities of, you know, talking to people and see what all I could do with my life. So.
Can I just bridge on Tori's question, because I thought that was a really interesting one. You know, we talked, and I mentioned I've been to Bangalore, and I've been to
Yeah.
Mysore 3 years ago, and I did see a lot of women in science. So could you?
So I think, like, even my institute, we would have a lot of women, like mostly in the biological department, or like few departments which are mostly like women-centric. In physics department, you would have like 5 or 6. So people are doing PhD and everything is all good. But what happens to those women faculty after the PhD? So in my institute, in my department, we don't have a single female faculty. We have around 12 or 13 male faculty. In physics department, we have 2 faculties. One is about to retire, and we just have just 1 female faculty. And so that even I don't know. I think it's just a constant pressure to be there where your families, you don't want to relocate. So what I always say in an Indian scenario is, like, if you're doing, if a woman is doing great in science, either they are not married or they're divorced. So that's what I have seen. so that's what I have seen. I mean, from my personal, like, people around me, like, they do pretty well if they're all single, not married, or they are on the separated part. So I think after PhD is where you have to really make that choice, like, okay, are you going to be with your family, or are you ready to take that step outside and move around? So that's the problem I always see. Like, doing PhD is great, but what happens after that is, yeah.
How do you organize the way you present next? How do you do that? How do you determine where you're going to present next? Next occasion.
For the Fulbright?
Yeah.
Okay, so I didn't. So my daughter was here till June. So from June to August is when her school closed here, and that's the only time I could apply for some Fulbright options, and that's why I wrote. And since he was here, I took North Carolina as some place where I could explore or have some people around, because I don't have to travel alone. So we have this Fulbright colleges in priority list, where they have given a list of around 2,000 institutions within US where you could apply to. So those, like the minority colleges, technical community colleges, and few universities. So those universities are more given priorities for supporting the OIL of the lecture fund. So I looked at some of the, and since I was moving to Raleigh, I thought I could go within 3 or 4 hours journey, and I was going through the list, and I came through your colleges. So one thing what I did is, all the colleges, even though there were a lot of colleges in and around North Carolina, 5 hours, I looked at colleges which had the nanotechnology department, because that would make sense. Otherwise, I cannot go to a normal undergrad. Undergrad is okay, yeah, and just say about Nano Motors, it won't make sense. Like I also, I have gotten approval for Florida International University. So the problem is the August is from June to August is where you have most of the students out of colleges. So they told me to come in September, but that's when I go and get my daughter back, so I have to see what I could do with that. Another one is University of Massachusetts at Dartmouth, so they also have a biotechnology, a little bit of nano program, so I could go there and present my work, but I have not looked at most of the places. Like if you see, we also have a few of the colleges listed in Hawaii, but I think it's mostly full. I guess all the people want to go and explore, so.
The reason why I asked that is the guys at Loma Linda University, which is in California, they'll be interested in this, especially the stroke group. I think that is something that is very interesting to, because if you could deliver this, because when I said drill the hole, because a lot of times drugs can't get through the blood-brain barrier, so it's easier if you could get in there to deliver the ETP. That would be perfect. So that's a place. It's in California. So if you could,
like I think for us, one thing is we have, like if we want to get approved for the oil, we have this sort of miles like restriction that you have to go beyond the certain miles to go and apply for this lecture fund. Which is why they want to us to explore it, and I think they want to kind of get people to see what all we are doing. I mean, I think they're kind of looking at more the undergrad level or grad level. I mean, not really like, you know, for us to really explore our research options, but yes, I could look into that. I will look into that, sure.
Okay. Also, one of the last questions. Okay, in terms of estimation, how many of those nanoparticles do you think you will need to like deliver an effective dose of drugs?
So here we do this electrochemical deposition. So we are talking about one membrane would give us around 10⁵ number of motors. And what we do is after we take these nanomotors, we look at the milligram range. So we would say around, let's say 3 to 4 milligram. And we also kind of measure like what is the number of molecules of drug molecules, because most of the drugs would have an absorbance peak, and we would kind of quantify that. So I would say maybe like 3 to 4 milligrams is what I have seen from my lab, what they have used for the experiments. Now I don't know exactly one membrane would give how many milligrams, but I can write to you back. I can check.
Okay,
sure.
that's fine. Oh, sorry, Russ.
Do you have a correlation between how the genes are expressed and how it affects how the particular particle gets into the cell, for example, or into an area where you need it to function?
At least for the gene-based experiments, the sensing experiments that I showed, we have not shown any in vivo experiments. It's mostly the cell-based, because most of them have been done with the ultrasound, and that kind of limits its application to the in vitro experiments. But we have only looked at, because most of these, the gene expression, we have only looked at the fluorescence sensing method, and I don't think we have really looked at the gene expression. I don't think so. It's too much expressed.
And the purpose of the hyperthermia experiment was killing cells like cancer cells and those type of defective cells? Because I noticed you had a hyperthermia study.
Sorry?
You had a study on hyperthermia.
Yeah, that's my work. Yeah,
cell death.
yeah.
So it was the target for that was to kill cancer cells.
Yes. Because it's most like a localized heat increment, and also the cancer cells are more prone to the higher temperature than the normal cells. So that kind of helps.
Really? Okay. How much longer are you in the States?
For 1 more year. I go back next August 2020.
We have a coordination network that we do through community colleges, and we're funded to do that. So if you're interested in going to other community colleges which are outside of the state, we can put you in touch with Ray.
Oh, sure. That's really nice. Oh, thank you.
Just
Yeah.
something we can do for you.
Sure. Thank you so much.
And if you come back during the school year, we'll take care of your baby.
She doesn't say that. They just come with.
Yeah.
That's nice. That's nice. That's good to see.
So I want to
Oh,
thank you so much.
thank you so
Dr.
much.
Lexman, for coming to talk to us. You know, as we talked about in the beginning, a lot of times we teach students of all backgrounds, and the work that you're doing is so important. And I just thank you for bringing it to all.
No,
of us.
it's my pleasure. I mean, I had a great time interacting with all of you. Thank you so much. Thank you.